Good morning, dear students. I am Bharti Kathuria Sehgal from the SD Vidya School. We are continuing the chapter number 4, Reproductive Health. Today, we are going to do a very important topic of this chapter, infertility. Dear students, reproduction is the characteristic of living beings which permits the individuals to make replicas of their kind. Living organisms reproduce themselves using a molecular blueprint called DNA. Living organisms are able to reproduce themselves, but if they fail to do this, population will diminish and disappear as their members die from old age, diseases, accidents, predation, and others. It is a fundamental law of biology that living things can only be produced by other living things. Every living organism owes its existence to the reproductive activity of other organisms. Humans reproduce by sexual reproduction. So, both the partners need to be fertile in order to be producing children. So, today we'll be discussing about the condition called as infertility. Dear students, do not confuse sterile with infertile. Sterile individuals are the ones which are unable to produce gametes. So the condition like Klinefelter syndrome in human caused due to an additional copy of X chromosome, or Turner syndrome in which there is a lack of one copy of X chromosome that we generally call transgenders are not able to produce gametes are called sterile. While the individuals who produce gametes but due to any physical, mental, psychological, immunological or congenital reason is not able to produce children are called as infertile. There are techniques now available to help such couples called as ART, which stands for Assisted Reproductive Technologies. Assisted reproductive technologies include all measures involving the manipulation of gametes and embryo outside of the human body to help a couple conceive. Assisted reproduction involves two main strategies: fertility medication and assisted reproductive technology. Fertility medication involves giving medicine as ovarian stimulator, which may induce the ovulation or which may help in maturation or release of the ovum. While the assisted reproductive technology involves either intrauterine insemination or in vitro fertilization. The assisted reproductive technology program comprises the medical techniques to assist women to conceive. The types of ART program include in vitro fertilization, intracytoplasmic sperm injection, and other ART procedure. The basic steps in ART program cycle are: the first and important step is medical assessment of the treatment. The second is commencement for the ovarian stimulation. Then ultrasound scans are done, after which the egg retrieval and the semen collection are done. The next step is fertilization and embryo culture, after which the embryo is transferred, and then there is a pregnancy test conducted to confirm the pregnancy. Now we will be dealing with the different types of the assisted reproductive technology. The ART includes IVF, AI, GIFT, ZIFT, ICSI. Now let's discuss about them. IVF stands for in vitro fertilization. In vitro means in lab conditions. Like in vivo stands for in living condition if the fertilization is occurring outside the body. In lab conditions is called as in vitro fertilization. If there the sperm count of the male partner is low, or any other problem with a male partner, then there can be direct intrauterine insemination. The other procedure is gamete intrafallopian transfer called GIFT, or zygote intrafallopian transfer called ZIFT. There is another procedure called intracytoplasmic sperm injection called ICSI. Let's discuss in detail about each one of them. Assisted reproductive technology has helped millions of infertile couples around the world to achieve pregnancy when all other options have failed. But we need to understand that who are being helped by the procedure of assisted reproductive technology. ART is an option for women who cannot conceive naturally or cannot carry pregnancy to the term or have genetic disorders or their partners have some abnormal sperm or sperm count. There are two main types of ART: artificial insemination and in vitro fertilization. Although generally ART is safe, but there can be risks like preterm birth, preeclampsia, and emotional distress. Success rates are high, but they depend on many factors. The very important factor is the woman's age. With increasing age, the success rate decreases. Now let's discuss about them and compare ZIFT, GIFT, and ICSI. ZIFT, I told you, stands for zygote intrafallopian transfer, a technique in which a woman's egg is fertilized outside the body and then implanted in one of her fallopian tubes. While GIFT involves a technique for assisting conception by introducing mixed ova and sperm into a fallopian tube. Intracytoplasmic sperm injection is a technique for in vitro fertilization in which an individual sperm cell is introduced into an egg cell. Let's discuss their procedures one by one. Let's compare first of all the in vitro fertilization. In the in vitro fertilization, eggs from the mother are combined with the sperm in a laboratory dish, and the fertilization when occurs, the embryo thus formed is placed inside the uterus of the mother. The next is gamete intrafallopian transfer. As the name indicates, there is no transfer of zygote or embryo, rather there is a transfer of gamete. So, eggs and sperms are collected in syringe and the unfertilized eggs and sperms are transferred to the fallopian tube. Fertilization occurs naturally in the fallopian tube, and the embryo thus formed in the fallopian tube will naturally slip into the uterus and gets implanted. Zygote intrafallopian tube transfer includes, again, the combination of egg with sperm in laboratory, and the zygote in a single celled stage can then be transferred into the fallopian tube, from where it can naturally move on into the uterus. The intracytoplasmic sperm injection involves the removal of the single sperm from the man's semen, and this sperm is then injected into an egg in a laboratory condition. The zygote thus formed, or the fertilized egg we call, is then transferred to the woman's uterus directly. Like we discussed, in IVF, the egg and sperm are collected and fertilization takes place in a dish outside the female body. Embryos are then placed in the female's uterus directly, not in the fallopian tube. So do not confuse each other. Always understand that gamete transfer will involve transmission of the gamete, but IVF will involve transmission into the uterus, while ZIFT will involve the transfer of zygote into the fallopian tube. IVF involves combining eggs and sperm in a laboratory. ZIFT will involve the placement of the young embryo into the fallopian tube. The intracytoplasmic sperm injection is used for couples in which there are serious problems with the sperm. GIFT involves transferring eggs and sperm into the woman's fallopian tube. So the fertilization in case of GIFT occurs inside the woman's body. So look at this picture. The egg production is stimulated by hormone therapy. The egg is thus retrieved from the ovary. The sperm from the donor or from the father is then mixed with the egg and are allowed to fertilize. After fertilization, the fertilized eggs are introduced into the uterus directly and not into the fallopian tube. While in GIFT, that is gamete intrafallopian transfer, the sperm and eggs are placed into the fallopian tube. In ZIFT, the zygote, which may be in a 2-celled, 4-celled or 8-celled or morula stage, is transferred into the fallopian tube and not into the uterus. So, in GIFT, laparoscopically, the eggs are placed in the healthy fallopian tube along with the sperm. While in the ZIFT, the zygotes are placed directly into the fallopian tube either via laparoscopy or by catheterization, that is by a simple surgery. The next, ICSI. ICSI is the technique in which sperm is directly injected into an ovum in the laboratory conditions. Assisted insemination with husband's sperm is involved when the husband has a low sperm count. So sperms are transferred by catheter into uterus or fallopian tube. This is also useful when the sperm motility is low. In vitro fertilization involves fertilized eggs which are transferred into uterus or fallopian tube. GIFT is used for the unfertilized eggs and sperm transferred into the fallopian tube. In ICSI, sperm is injected into the egg. In ZIFT, the zygote or early embryo is transferred into the fallopian tube. Because assisted reproductive technologies are being utilized for helping the people to have pregnancies and have children. But there has to be a regulatory authority which can keep a check on this method. So, a bill was established by the national board, the state boards and the national registry to regulate and supervise assisted reproductive technology. It establishes the art banks to promote ethical practices. It proposes stringent punishment for those who attempt to control the offspring's sex, sell embryos or gametes. As of February 20, there are 517 art clinics under the National Registry of Art Clinics and Banks in India, according to the ICMR. Our laws permit legal adoption and it is one of the best methods for couple looking for the parenthood. That's all for today, students. Thank you. God bless you all.